investigadora del Instituto de Genética, quien va a moderar este webinar del proyecto Microtoxbol, exposición arsénico, interacciones en ambiente y efectos en la salud en el altiplano central de Bolivia. Tenemos el grato placer de contar con investigadores con mucha experiencia en esta temática y a quienes realmente doy un cordial saludo, en especial al doctor Jacques Gardon, a la doctora Karin Broberg y al doctor Philippe Gerard, quienes dentro de un momento nos van a dar justamente, les vamos a invitar a que den sus exposiciones. Pero primeramente, para hacer un poquito más protocolar nuestro webinar, porque considero que con la calidad de expositores que tenemos, es muy importante darle este protocolo a este webinar. Voy a invitar a la doctora justamente Noemí Tirado, quien es la jefe de la Unidad de Genética Toxicológica del Instituto de Genética de la Facultad de Medicina de la UMSA, y además es presidenta de la Asociación Latinoamericana de Mutagénesis, Carcinogénesis y Teratogénesis Ambiental, para que dé las palabras de bienvenida a este webinar. Adelante, por favor, doctora Tirado.
Bueno, muchas gracias, doctora Cortés. Un saludo a los invitados que nos están visitando de Francia y de Suecia, al doctor Jacques Gardon, con quien trabajamos hace bastante tiempo. De igual manera, a la doctora Karin Broberg, que también estamos ya bastantes años con la cooperación. Y dar la bienvenida a Philippe Gerard, que es una persona, un investigador que se está integrando a nuestro proyecto. Así, de igual manera, quiero saludar al doctor Mauro Constantino, que nos acompaña, jefe del Departamento de Investigación y Posgrado Interacción Social de la UMSA. Y bueno, a todos los participantes por estar en este momento con nosotros para poder escuchar estas dos charlas y también una introducción que va a hacer el doctor Gardón de lo que es el trabajo que estamos realizando hace bastantes años. Muchas gracias, muy bienvenidos y espero que sea muy buena la participación también de las personas que están como asistentes y puedan hacer algunas preguntas. I don't know if I can speak a little bit in English because Dr. Karin is she speaks English, but maybe
Me too.
only to, also Philippe speaks English and French.
Yeah.
So we are very grateful to have this webinar with you, and feel free to talk, and we are going to translate if it's necessary. Okay, thank you, and we can start with the webinar.
Muchas gracias, doctora Tirado. Bueno, creo que es importante resaltar que este webinar está siendo organizado por el Vicerrectorado, por el Departamento justamente de Investigación, Posgrado e Interacción Social conocido como DIPGIS, y el Instituto de Genética de la UMSA, ¿no? Así que también doy un cordial saludo al doctor Mauro Constantino, ¿no? Que nos está acompañando también en esta oportunidad, director del DIPGIS. Bueno, voy a invitar a nuestra directora del Instituto de Genética, que es la doctora Jessica Barrón, para que ella también dé unas palabras de circunstancia, por favor, en esta oportunidad. Adelante, doctora Barrón, por favor. Creo que no está presente.
Sí, parece que tenía problemas de conexión. Está ingresando recién.
Ya. Entonces, si podemos esperar un minutito, por favor. Si está ingresando Jessica, que es la directora del Instituto de Genética, por favor. Que a veces estos problemas de conexión realmente interfieren, la verdad que, ¿no? En este tipo de actividades virtuales, ¿no? Así que, por favor, un momentito de espera.
Hola, sí,
Ahí está.
creo que eres tú.
Sí, adelante, por favor, doctora Barrón.
Muy buenos días, buenas tardes. Muchas gracias por la invitación. Agradecer a todos los invitados que se encuentran presentes, agradecer a la doctora Karin Brovery, PhD, docente de la Universidad del Instituto Karolinska, allá en Suecia, al doctor Philippe Gerard, también que es de Francia, al doctor Jacques Gardon, que es un arduo colaborador de hace muchos años con la colaboración francesa, a todos los presentes, a agradecer también al doctor Mauro Constantino por su colaboración, a Cristina Mejía también por su colaboración y por hacer posible esta transmisión. I will talk a little bit in English. Thank you so much to Karin Brovery for everything, for all the collaboration with this webinar, also with this all the contributions with projects and everything with Arsenic, and all the contribution with the Microtoxbol project. And also thank you so much to Philippe Gerard, who is now in collaboration with this Microtoxbol project. And it's a pleasure to meet you again and to thank you. I have to say thank you for this small talk, but I think it will be very, very, very helpful for everybody. And also I want to say thank you to Jacques Gardon. He's I can say he's another Bolivian person because he loves Bolivia and he collaborates a lot with many projects. Thank you so much. And thank you to Mauro Constantino also and Cristina Mejia from DIPGIS for make this collaboration possibility. And yeah, thank you so much. And we can start, I think, in the name of the Institute of Genetics. And we can start with this webinar. Thank you so much and very welcome to Bolivia again.
Muchas gracias, doctora Barrón. Y ahora sí vamos a invitar al doctor Mauro Constantino para que por favor dé las palabras de inauguración de este webinar. Adelante, doctor, que es el director del DIPGIS.
Muchísimas gracias. ¿Me pueden escuchar bien?
Sí, le escuchamos.
Sí.
Muchísimas gracias, doctora Cortés. Gracias a los participantes, a Noemí por la organización y obviamente a la directora del Instituto de Genética por las palabras. Es un placer recibir esos tipos de colaboraciones. Estamos muy convencidos como Departamento de Investigación, Posgrado de Interacción Social, de que la investigación es un asunto de redes, es un asunto plural. Y ese tipo de participaciones con investigadores de otros países que, como subrayaba quien me ha antecedido, además aman a nuestro país, creo que es fundamental. Es un placer poder cooperar en este tipo de organización. Dr. Karin, Dr. Philip, Dr. Jacques Gardon, it's really a pleasure to welcome you to Bolivia and to these talks. I would like, on behalf of our Vice Dean, Maria Eugenia Garcia, and on behalf of our department, to thank you for your participation both in research and in formation. We do believe that these talks can strengthen our capacity in research and most of all in the socialization and communication of science. We many times find out that it is one of our weak points, I would say, the socialization of our results, and we are starting a line of improvement in this sense. So your talk is really important, and we would love to for our community to participate in that. That's why we are trying to socialize that with also our social medias. It's really a pleasure to welcome you here, and again, thank you for participating in our efforts in research and socialization. Muchísimas gracias a todos los participantes y queda inaugurado este webinar. Muchísimas gracias. Adelante.
Gracias, muy gentil. Ahora vamos a invitar justamente al doctor Jacques Gardon, ¿no? O sea, es de la conferencia sobre antecedentes e
¿Me
introducción
escuchan? Porque
del
tengo la
proyecto
impresión que se cortó, ¿no?
Microtoxbol, exposición
No, creo que
arsénico, ambiente y efectos en la salud en el altiplano
vemos
central de
que...
Bolivia.
¿Me escuchas,
Adelante,
Philippe, no
doctor
ve?
Gardón.
No. Philippe, tu m'entends, non?
Oui.
Oui.
Sí,
¿Está
le
bien?
escuchamos, doctor.
¿Ya está bien?
Escuchamos.
Porque está sin movimiento, Jacqueline.
Creo que tiene problemas de conexión.
¿Se escucha? ¿No?
Sí.
No, está full. Sí, yo puedo empezar, ¿no ve?
Sí, usted, doctor. Adelante.
Escuchamos.
Usted empieza, doctor Gardón.
Ya, ok, entonces, vale. Entonces, muy buenos días a todas y todos. Gracias a la doctora Noemí Tirado y a Jessica Barón por la organización de este webinar. Quisiera introducir de manera breve el tema que tratamos hoy. Hace mucho con Noemí Tirado y su equipo del Instituto de Genética trabajamos sobre estos efectos de la contaminación ambiental por elementos tóxicos. La elaboración fue en los años 2007 a 2009 en un proyecto en Oruro, la contaminación minera y sus efectos sobre las mujeres embarazadas. En este momento quisiéramos evaluar el riesgo para la salud de la contaminación provocada por los elementos que están presentes en los compuestos de la mina. Después de considerando la especificidad del medio ambiente andino, profundizamos el estudio de exposición a un elemento que estoy resaltando, pero que es muy importante para la salud humana, que ha sido descrito bastante bien en Argentina, Chile y otros países de la cordillera, y no tanto en Bolivia. Estoy hablando del arsénico. El elemento que es muy tóxico puede encontrarse en agua donde hay contaminación minera, es decir, de manera orgánica, pero también en aguas de fuentes naturales, en particular en el contexto volcánico de la cordillera occidental. En el sur del lago Popó, la contaminación de las aguas ya había sido descrita por el equipo de María Eugenia García, que todos conocen bien, del Instituto de IQ, y otros colegas. Yo estoy hablando de Mauricio y Osvaldo, que trabajaron muchos años. Nosotros nos interesamos en la parte de exposición humana. Profundizamos el tema encontrando comunidades donde la toma de agua puede ser un contexto natural, población a este contaminante. Eso fue el punto de partida de un estudio apasionante por la cual se juntó un equipo multidisciplinario coordinado por la doctora Karin Broberg del Karolinska Institut, que nos va a hablar después. Nos presentamos resultados, hemos encontrado resultados impactantes. Sin embargo, las tesis que hemos tenido, se van a ver en la presentación de Karin después, no explican todo lo que observamos. Así hemos querido abrir un nuevo tema. Este nuevo tema habla de la relación que existe entre la microbiota del intestino y el medio ambiente. Es por eso que se juntó al equipo el doctor Philippe Girard, que es un especialista de este tema, de su relación entre microbiota y salud humana y enfermedad crónica. Y más lejos es a ello de presentar la parte científica, entonces dejo la palabra a la doctora introducir a nuestro colega Philippe Girard. Muchas gracias.
Thank you, Jacques, for this introduction, and thank you, Noemi, and all the organizers for inviting me to give a talk today. And I want also to say hello to Karin. Unfortunately, I'm not with you. I was supposed to be in La Paz today, but I had to cancel my travel, and so I give my talk from Francia. Today, I won't talk about our project because I knew in the Arsenic project, but I will talk about what I know the best, the microbiota. I will share my screen. I think now you can see my screen, I hope so. And so I will talk about the role of the gut microbiota in obesity and liver diseases, which is my scientific field. And I will particularly focus on the way we can use germ-free animal models to prove the causality between the gut microbiota and this pathology. Okay, you probably know that the gut microbiota begins colonizing the gut at birth in mammals. And so the first bacteria to colonize are the aerobes and facultative anaerobes, so the bacteria that can tolerate oxygen. And then the strict anaerobes colonize later, so the bacteria that cannot tolerate oxygens. And those first colonizers arise mainly from the mother vaginal and fecal microbiota, and also from the skin microbiota in case of cesarean birth. And so in the first months of life, the gut microbiota is not stable, it changes continuously, and a stable and complex adult-like microbiota is reached at around the age of 3 years, and after that, it's more or less stable until the age of adult. So here you can see the human gut and the bacterial density living in the different compartments of the gut. So I don't want to bother you today with the name of bacteria, but what is maybe more important is the bacterial population living in the different part of the gut. So we don't have a lot of bacteria in the stomach. It's not very good for bacteria. It's acid and they don't like that. So we have few bacteria in the stomach. But the more you go forward in the gut, the more the bacterial density increases to reach a maximum in the colon where you have up to 10 to the 11 bacteria per milliliter. And to give you a more striking image of what represent all these bacteria, we have more cells in 1 gram of feces than in a human brain, and as many bacteria in our gut than eukaryotic cells in our body. And not the same for everybody. We don't have the same bacteria. Actually, it's true for the type of bacteria that are present, the different species, but it's true also for the genes. Now we know that we have around 540,000 genes per microbiota, so it's many more genes than in the human genome, which is 20,000 genes. And half of these genes are shared by at least half of the population, so we are all rather similar in terms of microbiota, but we have also rare genes, so genes that are shared by less than 20% of individuals, and more than 2 million of these genes have been now identified. So we are not identical, we all have different microbiota. And these rare genes are very important because, for example, if you have the genes that are able to detoxify arsenic, okay, you can resist against arsenic. If you don't have in your microbiota, you will be more sensitive to arsenic. Some example of the function of the gut microbiota, what we call the barrier effect, is the capacity of the microbiota to protect from the colonization from other bacteria, including the pathogens bacteria. The gut microbiota is completely required for the maturation of the immune system. They produce essential vitamins, particularly K vitamins. This is the main source of K vitamin, but some bacteria in the gut also produce some B vitamins. And they metabolize environmental chemicals. I already talked about that for arsenic, but almost all chemicals that we can absorb can be metabolized by one or two or different bacteria. And so it's important because it can change the health effect of this chemical, sometimes in a good way, sometimes in a bad way. If some molecule, for example, drug, that can be inactivated by the bacteria, by the gut microbiota, they are no more active, and they are no more active. And more recently, it has been shown that the gut microbiota can influence the brain and also the energy harvesting and fat storage, and it could be in that way involved in obesity development, and this is what I will show in the next part of my presentation. So dysbiosis, dysbiosis is an alter microbiota, and actually it has been found in associated with a lot of diseases. So you analyze the gut microbiota from patients suffering from obesity or, I don't know, allergy or autism, and you compare to a healthy microbiota, and you find this dysbiosis, this alter microbiota. But it doesn't tell if the dysbiosis is the cause or the consequence of the pathology. So how can we prove that the gut microbiota plays a causal role in disease development? And I will show you in my presentation that we can use germ-free animal models and gut microbiota transplant to try to prove this causality. So in this slide, you can see a letter from Louis Pasteur. So it's written in French and it's very small. So I translated a few sentences. So in that letter, Louis Pasteur, 140 years ago, writes, "If I had the time, I would try to feed animals from the birth with complete sterile food." So sterile, he thinks, with no bacteria. And I have the preconceived idea that in this condition, life would become impossible. So he had the idea that we cannot live without bacteria. Nevertheless, he wrote, "If I could develop this system, we could try to study the digestion by adding single of a mix of microbes." This is really what we do in my lab now. And finally, whether life would be impossible or in contrary easier, it would be of great interest to do the experiments. So more than 100 years ago, Louis Pasteur had the idea that the creation of germ-free animals, so animals devoid of bacteria, would be a very nice model to study the role of the gut microbes in health and disease. And his idea becomes true 50 years later in the 30s in the US. And here you can see now a photograph of our germ-free animal facility in our lab in Jouy-en-Josas near Paris. Actually, this is the second oldest germ-free animal facility in Europe, and probably, well, I don't know, but if Karin knows, but the oldest germ-free animal facility in Europe has been built in the Karolinska Institute in Stockholm, but I've been told that it doesn't work anymore, so maybe we have now the oldest in Europe, and probably the biggest one in Europe. So what you can see here is called the isolators. So the isolators are the sterile chambers where we breed some germ-free mice and rats in our lab. So one can live without bacteria, or at least mice and rats can live without bacteria, and now I will show you how we can use these germ-free animals to study the role of the gut microbiota in health and disease. So the first way to use these germ-free animals is to compare them with conventional mice. So we compare animals with and without microbiota to see what is the impact of the gut microbiota, and it has been made particularly in the context of obesity and metabolic diseases. Here you can see that the germ-free animals are resistant to diet-induced obesity. So what you can see here is the body weight gain of animals with and without gut microbiota when given a high-fat diet. So animals with a gut microbiota become obese, and animals without a gut microbiota, so germ-free animals, they don't become obese, they stay lean. This is the first study showing that in the US, and on the right, this is a study from our lab. And in our lab, we have also shown that the germ-free animals did not develop hyperglycemia or hyperinsulinemia in response to a high-fat diet, so they do not develop insulin resistance. So germ-free animals, so animals without a gut microbiota, are resistant to obesity and insulin resistance. But okay, it's not real life. In real life, we all have a gut microbiota, so Okay, they are resistant to metabolic disease, but the main question is, do different gut microbiota differently impact obesity development? Or in other words, are there some gut microbiota that can aggravate obesity or produce obesity, or are there some gut microbiota that can protect against obesity? And to answer this question, we can transfer different gut microbiota to germ-free mice, and if it's true, we should get a different phenotype. And this is what has been done. So people often ask me how we do the gut microbiota transplantation. So I had a few slides to show you how we transplant some gut microbiota to germ-free mice. When we do a transplant from human to mice, you take some human feces. When it's from mice to mice, we prefer to take the cecal content. And you can see here the cecum of the mice because it's more liquid, it's less dry than feces in mice. And also you can keep the anaerobic bacteria because you can transfer this cecum, which is green here. It's green because this mice received a diet with a green dye because it's a high-fat diet. We have a green dye, so all the gut become green. So you take the cecum here, you transfer in an anaerobic glove box, so you don't have oxygen, you can keep the anaerobic bacteria, you prepare some suspension, and then you transfer this suspension inside the isolators, so the sterile isolator where are the germ-free mice. And here you can see Aurelia, a technician from my lab, inoculating the cecal suspension to the germ-free mice by gavage, and after 2 or 3 weeks, you have in the germ-free mice the same gut microbiota that you have in the donor mice. Okay, so in this kind of experiment have been done using transplantation of the gut microbiota from obese mice or lean mice, and as you can see here, when you transplant the gut microbiota from obese mice, the increase in body fat is twice higher than when you transplant the gut microbiota from a lean mice. So clearly, there are some gut microbiota that can trigger more obesity, more weight gain than others.
All chemicals that we can
same kind of experiment have been done with
metabolized by
a microbiota altered by low dose of penicillin. So in that experiment, the donor mice received a controlled diet or low dose of penicillin, so which alters the microbiota, and then the microbiota of this mice have been transferred to germ-free recipients, to germ-free mice, and the recipient mice did not receive antibiotics, just a normal diet. And as you can see here, the germ-free mice that received the altered microbiota gain more weight, and it was particularly true for fat, so the altered microbiota is able to also to increase the body weight and to trigger obesity. And it's also true for human microbiota. So in this study, the microbiota from 2 twins, so they were real twins, but discordant for obesity. It means that only one twin was obese and the other one was not. So they transplanted the gut microbiota from the 2 twins, so the obese twins and the lean twins, to germ-free mice, and the increased total body and fat mass was obtained only after the transplantation of the obese twin microbiota.
Bueno, muchas gracias, doctora Cortés.
Okay, now I will switch to some projects from my lab regarding the role of the gut microbiota in liver diseases. And as you will see, we use the same kind of strategy using germ-free animal models and gut microbiota transplant.
Bastantes años con la
So
cooperación.
the idea was to prove that the gut microbiota plays a causal role in liver disease, and how by transmitting different susceptibility to these two pathologies. One is a non-alcoholic fatty liver disease, and the other one is an alcoholic liver disease, via microbiota transplant to germ-free mice. So the first pathology is the non-alcoholic fatty liver disease. So this is a pathology covering different stages. The first stage is a steatosis, which corresponds to a triglyceride accumulation in the hepatocyte, so it's a fatty liver. So at this stage, the liver still works normally. But in a fraction of obese people with steatosis, an inflammation occurs. And so you have the NASH, the non-alcoholic steatohepatitis, which occurs in 20 to 40% of the patient, associated with insulin resistance, and sometimes it could evolve to fibrosis, cirrhosis, or colon or liver cancer, and in death in 10% of the cases. So this is alcoholic liver disease. So as you can see, the different stages are quite similar to the non-alcoholic liver disease, but here the steatosis is due to alcohol, so to heavy drinking. So long-term heavy drinking leads to steatosis, similar to obesity. This step is reversible after 4 to 6 weeks without alcohol. You can come back to a normal liver, but sometimes also it can evolve to a steatohepatitis, to a fibrosis, to a cirrhosis, and these steps are not reversible, and they can also lead to liver failure, portal hypertension, hepatocarcinoma, and the patients with a severe form of alcoholic liver disease have a very high rate of mortality, so it's really a bad disease. And so our question was, why only a part of obese and heavy drinker develops the severe stages of this pathology? Actually, it's not correlated to the degree of obesity, with the degree of alcohol consumption. So you can be a very high heavy drinker and stay all your life at the steatosis stages. And so it's not really understood why 20% of the patient go to these severe stages of the pathology. And so our hypothesis is that the gut microbiota could be a response to explain why only a fraction of these people develop these severe stages. And so in our first project, we do an experimentation only in mice, and I will now present this project regarding non-alcoholic fatty liver disease. And so this project comes from this observation. When you give a high-fat diet to conventional mice, so the conventional mice are the mice with a microbiota, but they, like in humans, they all have a different microbiota. You get heterogeneous responses. I mean that some mice become obese, some others do not. Some develop insulin resistance, some others do not. Some develop liver inflammation, some others do not, etc., etc. And our hypothesis is that they respond differently because they have a different gut microbiota. And to test this hypothesis, our strategy was to transplant the gut microbiota from 2 mice with opposite response to a high-fat diet to 2 groups of germ-free mice to see whether we can transmit a different susceptibility to diet-induced non-alcoholic fatty liver disease by transplantation of different microbiota. So the first step of the experiment was quite simple. We gave a high-fat diet to 24 conventional mice for 16 weeks, and we evaluate the parameters linked to the non-alcoholic fatty liver disease. So the development of obesity, of hyperglycemia, insulin resistance, hepatic lipid metabolism, liver inflammation, etc., etc. And as I told you before, we get heterogeneous responses, which means that some mice become obese, develop insulin resistance, inflammation, etc., etc., and some others do not. And so the goal was now to select the two donor mice for the microbiota, so the mice that developed the worst disease and the mice which did not develop any parameters of the disease. So to be more explicit, I show you here just 3 parameters that we have measured: the body weight, so the mice are ranked by body weight, the UMSA index for insulin resistance, and a cytokine as a marker of inflammation. We had more than 30 different parameters, but I just show you these 3 to be more simple and to be more easy to be understood. So each mouse is in the same column, so we have the 24 mice here, and from all these parameters we have selected these 2 mice. So one mouse that we call the responder mouse. So this mouse developed all the metabolic dysfunction, all the metabolic disorders, so high level of insulin resistance, inflammation, steatosis, liver inflammation, etc., etc. And the non-responder mouse, which did not develop any of the metabolic disorders, even if they received the same high-fat diet during 16 weeks. And as you can see, actually, these 2 mice were the same weight, which shows that you can be the same weight and have the pathology or not. And so the next step was to transplant the gut microbiota from these 2 mice to 2 groups of germ-free mice to see whether we were able to transmit a different susceptibility to non-alcoholic fatty liver disease. So the non-responder and the responder recipient then received a high-fat diet, and now I will show you the results that we obtained in the recipient mice. So again, the only difference between these 2 groups of mice was the gut microbiota that we transplanted to them. And so we found no differences in weight gain, but it was expected because the 2 donor mice were the same weight. And when we look at the glycemia, we showed that only the mice that received the responder microbiota develop an hyperglycemia, an hyperinsulinemia, and so the UMSA index for insulin resistance was found more than 2 times higher in the mice that received the responder microbiota compared to the mice that received the non-responder microbiota. When we look at the liver, we saw that only the mice with the responder microbiota developed a massive steatosis, and it was confirmed by the histological score, which also showed some inflammation only in this group, and it was actually associated with an increased expression of genes involved in the lipogenesis in the liver of this mice. So it means that the responder microbiota is able to send a signal to the liver to synthesize new lipids. Okay. So I won't go further for that project, but as you can see, we were able to transmit different susceptibility to non-alcoholic fatty liver disease by the transplant of different gut microbiota. Now I switch to the second project regarding the alcoholic liver disease. And in that case, we had a part with the human patients. So we have 2 objectives. One was to characterize the gut microbiota composition of chronic alcoholic patients at different stages of alcoholic liver disease. So you have to keep in mind that all the patients were alcoholic, but at different stages of the pathology. And again, to determine if we can transmit the different susceptibility to alcoholic liver disease via the human microbiota transplant to germ-free mice. So in this study, we include patients with excessive alcohol intake, more than 80 grams per day, with no antibiotic therapy, and the liver injury was assessed using a liver biopsy. So using this liver biopsy, a score was determined using anatomopathology and also using the number of immune cells in the liver biopsy. And from this characterization, the 37 alcoholic patients were divided into 3 groups. So a group with a severe alcoholic hepatitis, so it's a very severe and advanced stage of the pathology. A group with a non-severe alcoholic hepatitis, so they have an inflammation in the liver, but it's non-severe. And a group with no hepatitis, only a steatosis. So here you have these 3 groups. So in green, the group with no hepatitis. In blue, the group with a non-severe hepatitis. And in red, the group with the severe hepatitis. And I won't go into all the results, the characteristic of this patient, but I just want to focus on 2 of them. The first one is the alcohol consumption. And as you see, the group with the more severe alcoholic hepatitis consume less alcohol than the other group. So it again shows that the severity of the pathology is not correlated to the quantity of alcohol consumed. But if you look at the bilirubin and Madre discriminant function, which are 2 markers of the liver injury, clearly the liver of the patient in red with the severe alcoholic hepatitis was the liver was more injured. The disease was more severe in this group, clearly. So we analyze the gut microbiota in fecal samples in this patient. So again, all the patients were alcoholic. So what you see, it's not the effect of alcohol, but it's the microbiota composition depending on the severity of the pathology. And as you can see, we saw a different gut microbiota only in the patient with the severe alcoholic hepatitis, so the group in red, with more enterobacteria, more bifidobacteria, more streptococcus, and less atopobium than the other group. So the alcoholic patient with the severe alcoholic hepatitis have a profile of gut microbiota different from the alcoholic patient with less severe form of liver lesion. But again, it doesn't tell if it's a cause or consequence of the pathology. So the next step was to transplant the gut microbiota from this patient to germ-free mice to see whether we can transmit again a different susceptibility to the pathology. So we choose 2 patients representative of their group, a patient, so an alcoholic patient with no hepatitis and an alcoholic patient with a severe alcoholic hepatitis, and we transplanted their gut microbiota to germ-free mice, and then we gave a particular diet, which is called a Liber de Carli diet, which is a semi-liquid diet with 3% of ethanol in the mice for 5 weeks, and it is supposed to be enough to trigger the first stage of the alcoholic liver disease. So now, what happened in the recipient mice? So as you can see here, this is the quantification of the inflammatory cells in the liver of the recipient mice. So again, the only differences between the mice are the microbiota that have been transplanted. And so as you can see, the mice associated with the microbiota from the severe alcoholic patient, severe alcoholic hepatitis patient, have many more inflammatory cells, many more inflammation in their liver than the mice that received the microbiota from the alcoholic patient with no hepatitis. This is also shown on these histology figures, and as you can see on the right, the liver necrosis score was also found 5 times higher in the mice with the bad microbiota. Okay, so because in humans, in alcoholic patients, the alcoholic liver disease extends out of the liver, but also touch different organs. We also look at different organs. And the first one was the intestine. And as you can see here, we had a higher gut permeability in the mice with the bad microbiota, which is also found in the human alcoholic patient with hepatitis, with a higher number of bacteria that translocate inside the intestine, and you see also an increased inflammation score in the ileum of these mice. We also found more translocating bacteria in the mesenteric lymph node with in the mice with the bad microbiota, and more inflammatory cells in the mesenteric lymph node, and it was even more obvious in the adipose tissue, in the visceral adipose tissue, which was much more inflamed in the mice with the bad microbiota, which correspond to what is seen in human patients with alcoholic hepatitis. So the inflammation extends not only in the liver, but also in different organs. So like in the previous project, we were able to transmit a different susceptibility to alcoholic liver disease by the transplantation of the gut microbiota. And I think I have finished. This is what I wanted to show you today. And so just a few take-home messages. So the germ-free mice are protected from diet-induced obesity and insulin resistance. Some gut microbiota, including from humans, can influence the development of obesity and associated metabolic disorders. Different susceptibility to liver diseases are transmissible by gut microbiota transplant, demonstrating a causal role for the microbiota in this pathology. So the study of the gut microbiota may lead to the identification of patients at high risk of liver diseases. Human microbiota-associated mice, or rats could be a good preclinical model to test drugs or food components targeting the gut microbiota, or also maybe arsenic resistance. And finally, gut microbiota transplant in germ-free mice could be used to demonstrate a role of the gut microbiota in other pathology. And finally, I want to thank all my collaborators. This is a photograph of my research team, and you have the name of the people from the germ-free animal facility, and on the right of the different collaborators and financial support for the project I have presented today. And thank you for your attention.
Bueno, agradecemos al doctor Philippe Gerard por su excelente y brillante exposición sobre el tema de la microbiota intestinal en obesidad y enfermedades hepáticas, evidencia en modelos animales libres de gérmenes. Gracias, muchas gracias, doctor Gerard, quien obviamente él es investigador en el Instituto Micalis, responsable del grupo AMIPEM, Microbiología Alimentaria e Intestinal para la Salud Humana. Bueno, ahora sí vamos a invitar a la doctora Karin Broberg, quien es profesora en el Instituto de Medicina Ambiental en el Instituto Karolinska de Estocolmo, Suecia, y ella nos va a hablar sobre los mecanismos biológicos para tolerar el arsénico en el medio ambiente. Adelante, doctora Broberg.
So thank you very much, and thank you, Philippe, for an excellent presentation. Very exciting. I have a lot of questions, and but I think we will have to take them afterwards. So now we switch to humans, but I will end with bacteria in the end of my talk. And the title is Mechanisms to Tolerate Arsenic. And this is a collaboration with people in Lund, Karolinska, and also Argentina and Bolivia. So let's see if I can move this. So arsenic in drinking water is a problem in many countries of the world. And this is a map, which is a bit old now, that shows the probability of arsenic in drinking water above 10 microgram per liter, which is the WHO's guideline for concentrations in drinking water. And as you can see, in South America, there are quite a few areas with red, with elevated drinking water. Here is approximately 140 million people affected. I would guess that it's up to 170 million now with the more recent data. Apart from drinking water, it occurs in rice. Also, we can use it for cancer treatment very efficiently for some cancers at a high dose. Earlier, it had been used a lot for wood preservatives, but also in insecticides. So this is a picture showing the sources of arsenic. And you have to remember that there also exists organic arsenic in fish, seafood, algae, and bivalves. And they are, I would say, much, much, much less toxic. I cannot say that they're non-toxic, but compared to inorganic arsenic, which we find in food, to some food, but predominantly in water, that's the toxic one. And the inorganic arsenic in its trivalent and pentavalent form is taken up very efficiently in the gut and metabolized by the humans, and we will come back also to the bacteria, and excreted in urine. And this is a quick process. But it's not complete, the metabolism. So in urine, you find both inorganic arsenic and the monomethylated metabolite MMA, remember that one, and the demethylated metabolite DMA. And as you can see here, the numbers below, that if you eat some organic, for example, from seafood, you can get enormous amount of arsenic in urine, but that represents the organic and non-toxic. So it's very important to know what you've eaten before in order to assess exposure and maybe poisoning in some cases. So the health effects of inorganic arsenic are many, and it's a very potent carcinogen. It's one of the most potent carcinogens in the general environment. So it causes skin cancer, lung cancer, bladder cancer, and maybe some other cancers, although the evidence is not so strong yet. One of the early signs of arsenic-related toxicity is changes in skin pigmentation and the so-called hyperkeratosis, which the photo here should show, which you can find on the soles of the foot or on the hands. It's also affect the lung and the cardiovascular system. There are recent studies showing now a link to both myocardial infarctions and other type of cardiovascular diseases. Liver toxicity, kidney toxicity, and impaired immune defense. And both children and adults are affected. So I will focus a lot on the arsenic metabolism. So this is an important slide. When we then take up arsenic in the gut, it's metabolized mainly in the liver, and we secrete it in urine. And the monomethylated arsenic, MMA, is the most toxic metabolite. It's more reactive to all kinds of proteins. And the DMA, the more DMA you have, the faster it's actually secreted in urine. So you want low MMA and high DMA in urine. So it has been known quite for some time that the main arsenic methylating or metabolizing gene and protein is the ASO3MT. And here is a structure of this protein. It has been found in bacteria and also in different animals. And it's Barry Rosen's group in Florida who has done a lot of functional and biochemical work on this protein. So the arsenic metabolism efficiency matters for susceptibility. This is a rather messy picture, but what it overall shows that the more MMA you have in urine, so the fraction in urine, the higher risk for different diseases. And this is a review picture showing that you have high MMA for all kinds of outcomes and a high risk for those outcomes. So how does it look then with the possibility to metabolize arsenic? It differs between populations. So here is the average of most populations where you have a bit less than 20% of MMA in urine shown as red. And MATLAB is in Bangladesh, where we have a lot of arsenic contamination. Taiwan is also an area with a lot of arsenic. But in the Andes, there was some recent or old papers from the 90s showing a very low MMA and a high DMA. So that brought me to the hypothesis that there may be some genetics that influence the differences between populations. So in 2008, me and colleagues from Karolinska Institutet went to the area where they have found efficient arsenic metabolism. And this is in the Argentinian Andes, in the village San Antonio de los Cobres, which is a similar altitude as La Paz or Altiplano, and has a natural exposure to arsenic from the volcanic bedrock. We compared to people living in Matla, Bangladesh, where they have a much more recent exposure, because they started to drill wells for drink groundwater that should contain less microorganisms that could cause diarrhea. Unfortunately, it also contained a lot of arsenic, this drinking water. So since the 70s, it's a big poisoning on the people in Bangladesh. And this is the first paper, or one of the first papers we did, where we took one polymorphism or several polymorphisms in the AS2MT gene as candidates, and we genotyped them both in Argentina and in Bangladesh. And we had then slightly different, or we had different exposure levels, but we could see a similar pattern of how the genotype were associated with fraction of MMA in urine, which you have on the y-axis. So the CC genotype to the right has the lowest MMA. But there was a difference in the frequency of genotypes. So in Argentina, 71% of the women carried the CC genotype, but only 16 in Bangladesh. This is the work of Karin Engström, who defended a thesis on this. This led us, of course, to the hypothesis if there could be an adaptation to arsenic in certain environments where the exposure have been for generations. And this is a collaboration with Uppsala, Lund, and Karolinska in Sweden. Before we go into the issue about adaptation, I just want to say that that we adapt to the environment is a well-known feature of plants and bacteria. Here's an example of the myrrh operon that many bacteria that live in mercury-containing environments have to be able to metabolize organic and ionic mercury. This is an example from plants, and where Arabidopsis can live with arsenic in the soil, but if you knock out two ABC transporter genes, they cannot. You get they die after a while. So these are examples of adaptations for bacteria and plants. So what did we know about adaptation in humans when we started? There were only a few examples. One of the first was actually to tolerate lactose from milk, which was discovered in Africa in 2007. And it was probably a strong selection to be able to drink milk, as it is very rich in nutritional factors. Other examples are digesting starch, when we started to go from hunters to farmers, living at high altitude, as shown in Himalaya, and also to resist certain diseases from Bangladesh. But adaptation to a toxic environment for humans was not known. But in the area of South America, there are some places where we know that people have lived with arsenic for a long time. These are the Colombian Chilean mummies from Chinchero community in Atacama, which have high levels of arsenic in the hair and tissues. And they have been living in this area for a long time, so it implies exposure to arsenic for many thousands of years. So we set up the hypothesis that there is genetic selection for arsenic-resistant genotypes among humans in areas with arsenic exposure for many generations. So how could this tolerance then be achieved? So we then returned to the arsenic metabolism, and The hypothesis is that if you produce less of the most toxic metabolite and also more of the metabolite DMA that makes you eliminate the arsenic faster in urine, that would be favorable. And we then based our first study on the Argentinian Andes around San Antonio de los Cobres, where people in this region, based on archaeological findings, have been living for several thousands of years. In this area, there is varying exposure to natural arsenic in drinking water, but some areas with very high. This is a source of the drinking water to San Antonio de los Cobres. And this is the river where today it's about 800 to 1,000 microgram per liter. But when we did the analysis, the people had the drinking water level of about 200 microgram per liter, and it was fairly stable. So how did we do? We did a field study, and we selected then based on interviews and measurements of metals in blood and urine. We selected individuals with a wide range in urinary fraction of MMA. And here you see colleagues of mine, Marie Walter and Gabriela Concha on the pictures. And then, so this is a study population. As you can see, the fraction of MMA in urine varied from 1.2 to 22. This was a study on 124 women from this village. And then we did a genome association study. We genotyped them for about 5 million individual polymorphisms in blood. And this is the results. So this is a plot of all the polymorphisms on the chromosomes that we measured. So each dot represents a certain polymorphism. And then we did association between fraction of MMA in urine, and we found a very strong association with place on chromosome 10. And when we zoom in on this region, as you can see in the bottom, it's many, many polymorphisms around the ASO-MT gene that are associated with both MMA and DMA. So this was good because we already knew that ASO-MT can metabolize arsenic. So we could, in this population, show that ASO-MT is the major gene for arsenic metabolism. And this gene has also been linked to arsenic metabolism in Bangladesh. So it's not only unique for, this is a study from Bangladesh. Then we did the selection scan, and we used 2 different methods. And we need then populations that are fairly similar genetically to the population of interest, but with a presumed lower arsenic exposure in history. We selected people from Colombia and Peruvians from areas where it's not known to be high arsenic exposure over time. And first we did population genetics just to see that they are not too different from each other. And here you can see our population from Argentina in pink, and the Peruvians in green, and the Colombians in purple. And the first picture to the upper left show that they cluster together as well in the other picture. So overall, they are fairly similar genetically. Then we did the selection scan, and then we zoom in on chromosome 10 around ASO-MT, which is the orange line here. And then we compare our populations with the Peruvians and Colombians, and we can see that the people from San Antonio de los Cobres, they differ, particularly in frequencies for certain polymorphisms in ASO-MT. And this is based on one test, local-specific branch length analysis. We also, this is just to compare that if you switch Peruvians versus Colombians and people from Argentina, you don't see the same signal. Then we did another test, and this is to see if certain genetic regions are very stable or kept together over time, showing homozygosity. And we found a weaker signal around ASO-MT, but if we zoom in, we can see that there is a long stretch of DNA that for each generation is very stable and not broken up by recombination in San Antonio de los Cobres compared to Peruvians. So both of these tests indicate that the populations were genetically very similar, apart from around the H3MT gene, where the people from San Antonio de los Cobres showed strong enrichment of genetic variants associated with an efficient and less toxic arsenic metabolism. And this, our first conclusion then, that we found selection for adaptation to arsenic, which was also the first example of human adaptation to a toxic environment. Then is the question, has this occurred elsewhere, or is it just in this area in Argentina? And then we come to Bolivia. So this is the same geological and geographic settings as in Argentina, where we were. And we did then a study around Popo Lake, where we identified that there were elevated arsenic drinking water levels and the people were exposed. And we invited people from the three major communities in the area, around Popo, Uru, and Aymara Quechua, to do a similar study as in Argentina. So this is the blood sample. We see Noemi here with her team, and also filling in questionnaires. How long have you been living here and what do you eat and drink? We created a map of the arsenic exposure in the drinking water in the area. And as you can see, it varies. And then we characterize their arsenic metabolism efficiency. And Aymara Quechua here are AQ and U is Uru. And what we can see that they had actually an efficient metabolism of arsenic with low MMA and in urine, and particularly among the Uru population. Then we did again, compared different indigenous populations in Northern and Central and South America to see if these populations from Bolivia overall differed from other populations. And they actually clustered very well together in brown markers here. So they are genetically similar, also with the Argentinian population from San Antonio de los Cobres. Then we did a selection scan, but now with 3 different methods. And what we can see is that in the Argentinian Andes, which is the upper panel, we have a peak around H3MT. And we also find the similar peak in Aymara Quechua and also in the Uru. And we see similar patterns, but a little bit different depending on test. So we conclude here that we found this selection peak around H3MT in both women from Argentinian and Bolivian Andes. So if we take one of the SNPs here, one of the polymorphisms, and you see the frequency across different populations, you can say the G allele is the most common in most populations, but in Aymara Quechua and Uru, it's the T allele, which is really predominantly. And it's a TT genotype that is associated with lower MMA in urine. And actually, we found the highest frequencies ever seen in the Uru population. So then we find that selection is a common phenomenon for arsenic metabolism efficiency in the Andes. But then is the question, has this occurred in other species? And now I must ask, how many more minutes do I have? So I don't talk too long. Okay, I try to be quick here. So, as Philippe mentioned, also bacteria can metabolize arsenic. So this is, but here it's called the RSM, which is the methylating enzyme in bacteria, which is then ortholog to H3MT in humans. We did an evolutionary study to look at how has this gene developed evolutionary over time. And we took a amino acid sequences from the databases from 134 species, and we had 150 sequences. And we found AS2MT in all kinds of kingdoms apart from plants and insects, I would say overall. And what we see, to our surprise, is that animal AS2MT in red clusters more closely to bacteria in yellow, both in one group and in a second group. They differ a little bit, the sequences, and they cluster in two groups. I will come back to that, why. So this is not what we would expect from the common evolution of different proteins. Just to compare, we took other metal proteins like ATPase pumps for calcium and copper, and we found Sarca, which is the calcium pump, that it fits the tree of life, meaning that animals cluster with fungi and far from bacteria. And the same for the copper pump ATP7A. So based on this, we proposed another type of evolution, that what we generally consider as the common tree of life. And that is that during several occasions during evolution, we propose here 5 times, we have actually taken up the AS2MT or corresponding gene in bacteria directly from the bacteria to either the fungus, or to humans, or to any other animal. And this indicates that, and this is what's called horizontal gene transfer, it indicates that it has been a very beneficial for the organism actually to adapt by taking up this arsenic methylating gene for arsenic metabolism during evolution. But these were sequences coming from the databases, and we don't know if they actually could methylate arsenic or not. So we took 50 of these predicted amino acid sequences, and we did biochemical analysis to see if they could methylate arsenic. And we took both from the group 1 and the group 2 because they are slightly different. And we had both bacteria, we had green algae, we had fungi, and etc. And by chemical analysis, we could actually show that, yes, they can indeed methylate arsenic. And I just want to point out here one gene or one organism, Faecolobacterium prausnitzi, which is very common in the gut. It is about 5% in some studies show that this is a major bacteria in the gut. And we could show that, yes, indeed, it can methylate arsenic. And another study had actually shown that if you transplant, just like Philip showed, if you transplant this bacterium to germline-free mice, it protects the bacteria from arsenic. And here we could add another evidence that, yes, it can methylate arsenic, this bacteria. So the conclusions are there are selection to achieve tolerance to arsenic, and it has occurred both within a species, which is humans, as we showed from Bolivia and Argentina, and between species, as I showed in the last part here, from bacteria to animals or to fungi. And our second conclusion is that humans carry 2 bacterial systems for arsenic methylation. One bacterium-derived AS2MT from group 1 incorporated in the human genome during evolution, and one from group 2 in the Faecolobacterium prausnitzi present in the gut microbiome. And with that, I would just highlight the collaboration we have had since 2015 as the Toxbol project, and recently what we call the Microtoxbol project, by including our colleague Philippe Gerard and Maria Teresa Alvarez from UMSA. And we will continue to see if people living in the Bolivian Andes actually have more of the Faecolobacterium prausnitzi maybe, and also if the interaction between the two systems for toxicity and protection. So with that, I would like to acknowledge my colleagues, Karolinska Institutet, Lund University, Uppsala University, Copenhagen University, UMSA, IRD in France, and the funders. Thank you very much.
Thank you very much, doctora Robert. Agradecemos realmente por su brillante exposición y ahora vamos a entrar a la fase de preguntas que tuviesen el auditorio que está presente. Si que quieren hacer preguntas a cualquiera de los expositores, por favor. O sea, ahí simplemente levanten la mano para poder darles la palabra. ¿Tienen alguna pregunta para cualquiera de ellos? Estamos en la ronda de preguntas. Mauricio Bachea ha levantado la mano. Mauricio, por favor, Bachea, adelante. Mauricio Bachea, ¿tienes la palabra? ¿Tienes la palabra? Por favor, si puede encender su cámara y el micrófono para que podamos escuchar qué pregunta quiere hacer aquí a cuál de los expositores. Bueno, no puede encender la cámara ni el micrófono. Tal vez escribir en el chat y, bueno, pedir a la doctora, tal vez no he tirado, se lo pueda traducir en inglés a los expositores. ¿Puede hacer, Mauricio, la pregunta por el chat, por favor, y decir a qué expositor, a quién está haciendo la pregunta? No sé si habrá otra pregunta para cualquiera de los otros expositores, tal vez. Pueden levantar la mano mientras han levantado también 2 personas. Hay 3 personas que han levantado la mano. A ver, vamos a ver aquí en el chat. Bueno, están las... Por favor, la siguiente persona que quiera participar, por favor, que haciendo una pregunta mientras Mauricio Machea está resolviendo su problema de conexión, por favor. Las personas que levantan. Noemí, adelante.
Escribió una pregunta ya ahí en el chat para Karin. He visto evidencias de chipayas.
Dice, perdón. Sí, ahí está. Tal vez tú lo puedes traducir para la doctora, por favor.
We saw this.
No, we haven't seen any signs of the skin changes, neither in San Antonio de los Cobres nor in any of the people in Bolivia so far. But this is I think maybe based on now 650 persons total.
Eso, y bueno, también con la otra parte de la pregunta que decía que, bueno, ellos piden, claro, soluciones. Justamente se está viendo, obviamente, si bien no presentan esa pigmentación o algún efecto nocivo para la salud en este momento, no es bueno que ellos consuman el agua contaminada. Entonces, bueno, dependiendo de los estudios que hagamos con una población mayor y viendo los lugares con ustedes también, Mauricio, trabajando en la parte de agua, se está viendo la forma de que se pueda plantear tal vez algunos proyectos que ellos puedan presentar a su municipalidad o ver algunas otras cooperaciones que les puedan ayudar a utilizar filtros o cavar de repente los pozos con otras profundidades para que puedan obtener el agua que no contenga el arsénico.
Muy bien, también hay Leonardo Roberto Rocabado. Hay una pregunta que también está en inglés, por favor, ahí si la pueden leer, está en el chat.
So, yeah, a little bit about the root or the phylogenetics of these genes. Yeah, it's a good question. We don't know from which bacteria or for which group of bacteria this gene actually originates. We found it mainly in we think it's a protobacteria where it has originated, but we are not sure when in time. You know that arsenic has been around in the environment for a very long time. So this has probably been very important in order for the bacteria to survive, particularly in environments with high arsenic. And the arsenic levels have also changed during evolution due to the great oxidation event. So very early during evolution, some systems have been established in order to handling the fluctuating concentrations of arsenic. And probably these systems have been efficient also for other organisms like eukaryotes, so that it has been beneficial for them to take up these systems from different types of bacteria. But exactly which bacteria it comes from and when, we do not know.
No tenemos otra pregunta en el chat. No sé si alguien más quiere hacer alguna pregunta, tal vez a los otros expositores. No sé, ahí está también el doctor Gardon, creo, levantando la mano. Adelante, doctor Gardon, por favor.
Muchas gracias, doctor Cortés. Tengo una pregunta que hacer a Felipe. Es pregunta del... Aquí. La gente que tiene disbiosis, ¿tiene síntoma o no tiene síntoma? Se puede, al preguntar a la gente...
Jacques, je parle pas espagnol.
Sorry. Yeah, I'm sorry. You show us evidence that
Thank you.
there is a strong relation between the bacteria and dysbiosis and the illness. We can have obesity and other things. I have two questions. My first one is, do these people have symptoms of this dysbiosis? I mean, but is it accessible to a physician by the questionnaire of the people to access this possibility of dysbiosis? This is the first one. And the second is, when will we be able to ask for a diagnosis in the laboratory, in routine, let's say?
So, okay, I can put again my camera.
You have us, Philippe?
Okay,
yeah.
Yes. So that's a very good question.
We cannot hear you. Well, is you here?
You don't hear me? It's open.
Yeah.
Okay. Okay, so that's a good question, but maybe my answer will not be as good, because it's very difficult to answer that question, because actually there are many factors that can impact the microbiota. The main one is the diet. So it means that for each pathology where you find a dysbiosis, you don't have a clear signature for each pathology. I don't know if you hear what I mean. There is a rule who say that each dysbiotic microbiota is dysbiotic in his own way. It means that, okay, the healthy microbiota, we see more or less what it is, but there are a lot of different dysbiotic microbiota. So you know that something is wrong, but you cannot see, okay, this microbiota is linked to obesity, this one is for diabetes, this one is for autism, etc. So it's very difficult. Probably with new techniques, we will be able in the future to do what you say. It means that, okay, maybe we have some markers if you have this bacteria or these metabolites produced by this bacteria, you have more risk to have diabetes or more risk to have this kind of pathology. But at that time, it's very difficult to diagnose a pathology with the microbiome. We have some exception. For example, for regarding liver disease, there is a paper showing that with the microbiota, you can predict with 80% the risk of cirrhosis, the presence of cirrhosis. So in some particular cases, it works. But you have to add the analysis of the microbiota in terms of bacteria, but also do some metabolomics to see some metabolites produced by the microbiota. So it's not that easy. It's not something that you can do, a physician can do in his cabinet.
Muy bien, tenemos también una pregunta en el chat de María Natiesda Ojero Valle, ¿no? Entonces, no quisiera, por favor, que se la traduzcas a esta la pregunta, por favor. Y que yo la leo en español o tú la vas a leer, Noé.
Sí, la que está en el chat.
Sí, dice, buenas tardes, quisiera saber.
Ya. Si el efecto del arsénico ha sido estudiado en grupos diferentes a las comunidades locales. Además de personas embarazadas y con obesidad, sucede que un familiar médico hizo prácticas en la mina de cobre y luego en un campamento minero, cerca del lago Titicaca. Su niña nació con problemas de corazón y sin una manito. Años después tuvo una niña normal. Hay estudios de esos casos. Nos puede causar... Bueno, eso es la pregunta.
Bueno, es complicado de contestar a eso, lo que acaba de decir Felipe. No hay evidencias, en muchos casos, entre metales y malformaciones. Y hay varios otros asuntos que pueden dar malformaciones. Así que no creo, no sé si que puede complementar la doctora Karen Robert. Yo no veo un metal relacionado con esta enfermedad. Hemos visto en los estudios que hicimos, en el corte que hemos tenido en Oruro, por ejemplo, ¿cómo se llama? Fanta palatina, ¿ya? En varios niños hemos visto, ya cada vez no había relación, no hemos logrado correlacionar, ya en la literatura tampoco se encuentran cosas evidentes entre metales y eso. Hay evidencia genética del riesgo de malformación, hay evidencia de otros contaminantes, pero no mucho de los metales.
Muy bien, muchas gracias, doctor Gardon. También Leonardo Rocabado tiene también otra pregunta, por favor. Ahí Noé.
Acepto Adaptation.
La pregunta de Leonardo Rocabado, por favor.
Sí. Perdón, ¿estás escuchando? ¿Se está escuchando?
Sí, se escucha, se escucha.
Ya, ahora sí.
Pueden responder.
¿Se escucha? Sí.
Sí, se escucha, Noé.
Ya, muy bien.
On n'entend j'entends pas Noémie, j'entends pas.
¿Cuál de ellos va a responder, Noé?
No, está respondiendo la doctora Broberg. ¿No se escucha?
Pero no se escucha, no se escucha.
Ah, perdón. Ya, ok, vamos a aumentar el volumen, pero sí está.
Porque a ti se te escucha bien,
Ya.
pero ella no.
Ahora sí.
It has been speculated on arsenic as being an electron acceptor in a paper by Barry Rosen and colleagues that did a similar evolutionary study as we did, and actually found the same results of horizontal gene transfer. So I recommend you to read that paper, and I'm happy to transfer or send it to you if you would like. While I'm talking, I have a question to Philippe, if it's okay. Yeah, Philippe, I was wondering, is it mainly through the immune system that the microbiota is exerting its effect on different organs? Or could it be totally different pathways? I mean, you have focused a lot on inflammation, and that's part of the disease. But if, for example, if you think about the nervous system diseases, think still at it, the link between the gut microbiota and autism, for example, is it still through the immune system, or is it something, what other pathways could be involved?
Yeah. Okay, so but also a very good question. So some of the effect of the gut microbiota are clearly through the immune system, but some other are completely independent of the immune system, and so it's sometimes difficult to understand how the gut microbiota can influence our brain, for example. But actually, it's easy to understand if you think that the microbiota produce hundreds of different metabolites or small molecules that are absorbed in the intestine and go to the blood, and then when they are in the blood they can touch every organ. So for example, for autism, in my team we have a project dealing with indole, which is a metabolite produced by the microbiota from tryptophan, and which could be involved in some brain problem, including autism, and it's completely independent of the immune system. And you have a lot of different examples of this kind of effect by some gut bacterial metabolites that are not linked to the immune system.
I think that now what you showed for obesity and diabetes, which are systems quite similar in mice and humans,
yes.
So you compare. But now when we enter and look at the interaction between the microbiome and chemicals, which are maybe metabolized differently between species, will be very complex but very exciting, because we know some species are very sensitive to chemicals. And others not. So that may be also the link to other systems than the immune system.
Yeah.
I'm looking forward to this.
I agree.
Muy bien, agradecemos a ambos expositores por la respuesta que han dado. No tenemos otra pregunta, por lo menos en el chat. No hay otra pregunta, excepto si alguien más quiere hacer alguna pregunta a uno de los expositores, puede levantar la mano, por favor, y vamos a dar opción a una pregunta más si es que la hubiese. Bueno, parece que nadie más va a hacer una pregunta. No nos queda más que agradecer a los tres expositores por sus brillantes exposiciones y el gran aporte que están haciendo en este campo de la ciencia. Y realmente ha sido, bueno, muy grato tenerlos acá en Bolivia. Espero que vuelvan pronto. Van a ser siempre bienvenidos. Así que muchísimas gracias también a todos los que han estado presentes, a todos los que han estado acá en este webinar, que su presencia ha sido realmente relevante y además que creo que nos hemos nutrido todos, ¿no? de estas últimas investigaciones, ¿no? en este campo. Listo, muchas gracias a todos, nos estamos viendo, que tengan un buen descanso, ¿no? Hasta luego, gracias a todos. Gracias a Cristina Mejía por el apoyo logístico.